A very good morning to faculty members and respected dignitaries present here.
I am
I am Pragati Singh.
a
A
very good
very good
morning to
morning to
faculty
faculty
members
members
and
and
respected
respected
dignitaries
dignitaries
present here.
present here.
I am
I am
Pragati Singh,
Pragati,
Assistant
Assistant
Professor in
Professor in
Pharmacy
Pharmacy
Department at
Department at
SRMU.
SRMU.
I
I
welcome you
welcome
all
you
in
all
our
in our
Faculty
faculty
Development
development
Program
program
2023.
2023.
The
The
topic for
topic
this
for this
program
program
is
is
Innovative
innovative
Research
research
in
in
Pharmaceutical
pharmaceutical
Industries.
industries.
To
To
share her
share her
valuable
valuable
knowledge and
knowledge and
enlighten us
enlighten
with
us
her
with her
experience,
experience,
knowledge, and
knowledge, and
exposure in
exposure
the
in the
field of
field of
pharmacy,
pharmacy,
we
we
have
have
respected
respected
dignitary
dignitary
present
present
among
among
us,
us,
Ms.
Ms.
Deepika
Deepika
Gupta
Gupta
ma'am,
ma'am,
who is
who
going
is
to
going to
present her
present her
views and
views and
experience on
experience on
topic
topic
drug
drug
repurposing and
repurposing and
innovative
innovative
approach in
approach
drug
in drug
research.
research.
Ma'am is
Ma'am is
currently
currently
working as
working
an
as an
associate
associate
professor in
professor in
Maharishi
Maharishi
School of
School of
Pharmaceutical
Pharmaceutical
Sciences.
Sciences.
Ma'am doing
Ma'am doing
PhD from
PhD from
Institute of
Institute of
Pharmacy
Pharmacy
Sri Ram
Sri Ram
Swaroop
Swaroop
Memorial
Memorial
University.
University.
Ma'am
Ma'am
completed her
completed her
Master of
Master of
Pharmacy and
and
Bachelor of
Bachelor of
Pharmacy from
from
TTC
TTC
Institute of
Institute of
Pharmacy,
Pharmacy,
Kanpur.
Kanpur.
Having total
Having
teaching
total teaching
experience 6
experience 6
years and
years and
4
4
months.
months.
Ma'am has
Ma'am
3
has 3
research
research
article and
article
1
and 1
review
review
article and
article
3
and 3
Indian
Indian
patent
patent
published.
published.
She is a
She is
life
a life
member of
member of
Association of
Association of
Pharmaceutical
Pharmaceutical
Teacher of
Teacher of
India,
India,
FT.
FT.
She
She
attended
attended
various
various
FDP workshops,
workshops,
seminars, conference,
conference,
national
national
seminars,
seminars,
and
and
international
international
seminars.
seminars.
Let's
Let's
welcome to
welcome to
Miss
Ms.
Deepika
Deepika
Gupta
Gupta
ma'am.
ma'am.
Ma'am, please
Ma'am, please
continue
continue
the
the
FDP
FDP
program.
program.
Okay. Hello everyone. Am I audible to you?
Yes, ma'am.
Ma'am.
Okay. Thank you for the brief introduction, Pragati ma'am. On the behalf of Maharshi University, I would like to welcome organizer of this faculty development program, Dr. Manju Pandey ma'am, uh, Dr. Shikha Srivastava ma'am, and the entire team of SRMU. Thank you for providing this beautiful opportunity. I also welcome all the participants. Hope this session is going to be informative. My topic is drug purposing and innovative approach in drug research. I chose this topic because as we already know that the faculty development program is it, and as you very well known that the faculty development program is about the innovative research in the pharmaceutical industries, and I thought the drug repurposing and innovative approach in drug research is an also a innovative uh, research. So I choose this topic. Hope you enjoy this session. So, what is the drug repurposing? As you, drug repurposing, also known as a drug repositioning, drug retasking, drug reprofiling, drug rescuing, drug recycling, drug redirection, and therapeutic switching. These all are like a synonyms of drug repurposing. But in this instead it reflects that all the words that either the drug repurposing, either the drug retasking or drug rescuing, the re word is repeated in very much. The re means the repeatedness of any use, any purpose, any kind of therapeutic indications that can be used, that can be utilized. Process of identification of new therapeutic use for old existing available drug, or we can say the FDA approved drug, or yeah, already marketed drug can be repurposed. Drug repositioning is an effective alternative approach to the traditional drug discovery process. We can say also, as we know, that traditional drug discovery process is somehow, we are very much familiar that it is a process of time consuming, labor consuming, and a long duration of action for the development of any drug. It also increase the risk of the failure. But in drug repositioning, it is an alternative as well as effective approach for the traditional drug discovery over the traditional drug discovery. It is an emerging strategy where the existing medicines are compact, particularly rare, or we can say difficult to treat or neglected disease. Some disease are also don't have drug to treat them. Then we can use we can use already existing drug in this repurposing approach, and we can utilize it for the new indication or new therapeutic approaches. Next slide. So now we can discuss about the traditional drug discovery versus the drug repurposing. As here is the flow sheet, and we can easily find out the benefit, the similarities, and the differentiation between the drug repurposing approaches and as well as the traditional approach. So, the traditional approach of drug discovery include the 5 simple steps like discovery and preclinical trials, safety review, clinical research, FDA review, and FDA post-market safety monitoring. And this is comes under the de novo identification of new molecular entity, which in which we includes all these 5 steps. But in case of drug repurposing, it includes only a 4 step, that is the compound identification and compound. Wait a while. The compound identification, compound acquisition, development, and FDA post-market safety monitoring. It is a low-risk failure and less time taken. As we compare, the traditional approach have the 5 step, and the drug repurposing approach only have a 4 step. Then we can say it is a low risk factor and cost effective, less time taken, and it is only happened because of advancement of bioinformatics and cheminformatics tools that are available in a huge biological and structural database. The traditional approach basically, as we discussed earlier, basically have include the 5 stages in drug discovery and clinical trials. That 3 step of first 3 steps are very much time taking. As you seen in slide, that the drug repurposing include the in silico approach, which is instead of drug discovery, clinical trials, and safety review. That in silico approach are AI tool, and we can also use a computational approach instead of these 3 steps. Then we can see skip this step. We don't require any kind of animal. We can say the chemicals. So it is a very cost effective approach. Now we discuss the strategy of drug repurposing. So there are 2 strategies. Basically, we can compare the strategies. One is a on-target strategy, another one is off-target strategy. On-target strategy is a kind of a strategy in which the drug is used for the known target, and it gives a known therapeutic indication as well as it gives a new therapeutic indication. When we analyze the new therapeutic indication, then we can claim for the repositioning of that drug. But in case of off target, we use the drug for the known target, and also we can use the drug for the new target. The known target and new target both are included in off target approach. Off target, we can say off target strategy. Then when these drugs are used, then known target gives a known therapeutic indication. If when we use for the new target and the new target gives new indications, then we can claim for the repurposing of this drug. The best example of the repurposing of on-target drug is minoxidil. This drug transformed from antihypertensive vasodilator to anti-hair loss drug. As an antihypertensive vasodilator, minoxidil has a property to dilate, or we can say has a property of widening the blood vessels and opening the potassium channel, which can increase the more oxygen demand and allow the oxygen to the blood and enhance the nutrition in the hair follicles. That's why it can be used for the treatment of male baldness. And the another example of off-target drug is aspirin is the best example for the off-target drug, as we know that the aspirin is an anti-inflammatory action used for the anti-inflammatory action. It is a NSAIDs and it is used for the various pain, but it also have a property to suppress the coagulation of blood. So by inhibiting the normal platelets functioning, so it can be used for the to treating the heart attacks, and also the new indication is also like a prostate cancer. It can be used for the prostate cancer treatment. Next, I discuss about the approaches of drug repurposing. As the slide reflects, there is a three cyclic phases, that is the in vitro, in vivo models, and the computational model. So, in vivo and in vitro models are as combinedly known as experimental model. And the in silico, or we can say the computational model, is a set of data from where we can identify, we can analyze which drug is useful for the repurposing purpose, and the entire database, entire structural information, all the information we can get from the in silico and the computational databases. So these are the two approaches. In vivo and in vitro models are the experimental models, and it is very much cost effective and time consuming, as well as labor consuming process, or we can say approach. In vitro process, we can need a culture media and several chemicals to find out where the drug is useful or not for that purpose. And in vivo model, we can use the animals, and we can use the organs of the animals and human subjects. So this is the cost effective cost effective approaches. But if you are go for the repurposing of any drug, you can use these three phases of cycles. If there is requirement, you can go for the experimental. You may or may not required for the experimental approach, or we can also repurpose the drug with the help of the in silico approach, or by the computational approach. So there is the approach. There is the classification of the approaches. So we can also we can easily understand the activity based approach and in silico approach, as we discussed earlier slide, that experimental approach include in vitro and in vivo screening. When we select any drug for repurposing or for any test, any drug development program, we can go through the experimental approach. If we try the activity based approach, then we can test the in vitro studies and in vivo screening. Then we go through the target based and cell organism based screening and assay. This approach do not require any structural information of target protein and drug-induced cell disease phenotypic information, but it is time-consuming, labor-consuming, and higher in cost. But its advantage is that the lower the rate of positive fall hits during the screening. In case of in silico approach, the computational screening can be done and protein target based screening can also be done, because some treatment required the protein alteration. So protein target based screening is very much required in this computational, or we can say the in silico based approach. It also requires the structural information of target protein and drug-induced cell diseases, phenotypic information. It is less time consuming, less labor consuming, less cost effective as comparison to activity-based approach. But the drawback is here, that is the higher the rate of false positive hits during the screening. So I suggest to the new researcher, if you go through these in silico based approach, you can study properly all the factors which going to be helpful during this development of drug or during the repurposing of drug. There is a in silico method, that is the molecular docking you can go through, and pharmacophore modeling you can go through for repurposing the drug for any kind of disease treatment, any new indication, for the treating our new indications. There is the methodology of drug repurposing. The methodology adopted in drug repurposing can be divided into the three broad groups. Depending on the quantity, quality, and pharmacological, toxicological, and biological activity information available. So these, we can say drug repurposing is based on the quantitative, qualitative, and pharmacological, toxicological, biological activity information with go through with these steps. But if we can say the methodology, then these are mainly three kinds. We can focus on the drug, or we can focus on the target, or we can focus on the disease or any kind of therapy. So if we go through the drug repurposing, the thought process behind the drug repurposing is that on focus on focusing the drug, or by focusing the target, or by focusing the disease. So this is the methodology of drug repurposing. In flowchart, we see the thought process. A thought box is a phenotypic or computational screen. It required the phenotypic or computational screen if you go through the drug repurposing. And this computational screen is very much kind of thought process required, like a new target, old indication, new target, new indication, old target, new indication. When you screen out that drug, then you may or may not go to the in vitro screening. You may or may not require the in vitro screening. If you require the in vitro screening, you go through the in vitro screening, then you go through the in vivo screening, then you can go for the clinical trial easily, and for the FDA approval, and for the successful, if you indicate, if you find any indication of new therapeutic responses, you can successfully claim for that, for drug, to claim for that repurposed drug, claim as a repurposed drug. And this methodology required less time, as we know that traditional approach is required approx 3 to 20 or 16 years about for the drug development. But by going through the repurposing approach, we can easily less easily short we can do it in a shortcut, and we can achieve the drug development in within 3 to 6 years, we can say. There are the reposition drugs. Sorry. There are some reposition drugs where the strategies are going to be like S1, S2, S3, S4, S5, S6, and S7. The methodology is based on drug. When the methodology is based on the drug, the methods and approach like a molecular docking, pharmacophore modeling, resources are VHTS and HTS, and reposition drug like uh, uh, thalidomide and bupropion. Then chemical similarities, if we go through the second strategy, we go, then the method is chemical similarities, fragment-based screening we can do, and the resources is genomic, and this is the truth. Then we go through the strategy 3. Then the methods and approaches are biochemical assay, cellular assay, animal model, and sources are proteomic, proteomics. Then this is the drug. These both drug are repositioned earlier. And the S4, if you go through the strategy S4, this is the gene expression, protein expression, metabolites, and the resources are transcriptomic, and the drug is these both drug are repositioned. And the strategy 5, uh, in strategies 5, that is based on the pathway based, this is the signaling pathway, omic data mining, these are the approaches. And resource is the metabolomic, and duloxetine is the drug. Then if we target the disease, we go through the strategy 6, and the method and approach is biomarker based assay, phenotype screening, and the resources are bioinformatics, cheminformatics, and the drug is drug which are repositioned, that is aspirin and metformin. When we go through the side effect, side effect based, that any drug we can take, and these and give their side effect, that side effect also give a treatment. Then we go through that strategy, and the method of approach are side effect, similarities, clinical studies, and the resources are databases. And the reposition drug are uh, sildenafil and minoxidil, as I gave earlier an example of minoxidil in uh, first two slides. These are the strategies of drug repositioning. Uh, and S1 is the accidental approach and strategy, and S2 is the observation of novel activity, like specific disease, phenotype, rational approach. And S3 is a new target interaction. S4 is the new role for existing protein target. S5 for new biochemical pathways, and S6 is a disease specific repositioning, and S7 is unexpected side effect are seen when any treatment is go through. These are some drugs and their origin. Sorry. So these are the status of development of these drugs, and it is used for their old indication and their new indication are present in this slide and this uh, uh, flow sheet. Then we can see many examples are there, like amphotericin B, antifungal and antibiotic drug that is used for the fungal infection, but it is also repurposed for the leishmaniasis and already developed drug. This is the already developed example of the drug. And the aspirin is a nested, as I interruption. So you can go through these slides, and there is many examples as you've seen in the slides. Some are underdeveloped and some are underdeveloped and some are already developed drug, and it as a repurposed drug we can say. uh, there are the underdeveloped drug are identified by the hashtag, and this already developed are defined as a streaks. So these are the examples of underdeveloped and already developed and important approach of repurposing drugs. They gives their old indication as well as new indication, and they are very helpful for the treatment of many diseases. So these are the opportunities and challenges. There we can say there are over 6,000 rare disease that lack a proper treatment. About 5% of them are being researched. So there are a huge number of diseases that don't have a proper treatment. So we can easily go that we can easily provide the treatment by if we go through that repurpose approach. It is less time consuming as a traditional as comparison to the traditional approach, and it's have a several benefits and several importance factors. Rare disease have a larger potential market to explore. If you are a researcher or if you have uh, working in an industry, then you can go through the target the disease, and the disease have a potential to market explore. If you target the disease, you can easily explore the market. Computational or machine learning approach has a significantly improved the performance of drug repositioning. In mixed approaches, computational methods are validated by the biological experiment and clinical test, as I earlier discussed the in vivo and in vivo in a 3 cyclic phases. Mixed approaches of repositioning offer a rational and executive exploration of all the possible repositioning opportunity, taking into the consideration improved access to the available database and technology advances. Various database and software are available publicly for genomic, proteomic. These are the resources by which we go through for the development of any kind of drug to repurpose. Uh, Likewise, metabolomic and pathway analysis. Several computational approaches already developed to increase the speed and ease of repurposing processes. Like as I discussed in earlier slides, some important database used in repositioning studies. There is I provide also a link of the website from where you can easily find out the database information available about, like you need information about the chemical structure. You go through the database like a PubChem, drug bank, Chem Spider, Chem DB, therapeutic target database, and these are the links I provide in the slide. If you add if you require the information about the target 3D structure, you go through the database like PDB and OCA or a Proteopedia, and these are the links. If you require the drug target information, you go through the drug bank, therapeutic target database, pharmacogenetic knowledge base, and drug map central, and these are the links you can easily find out this. Likewise, protein interaction information you get from the biological general repository or interaction database of interacting proteins or strings, and these are the links. And the pathway information, if you require, then you go through the NCI pathway interaction database. Likewise, pathway of commons and clinical trial information and adverse effect, you can go through the adverse selection database, Canada and CIDRS. These are the links for to access these databases. Then if you require the FDA label information, you go through the daily med or structured product labeling. And if you want omics database that is based on the target or drug, you go through the NCBI, GEO, and sequence read archive, array, express, cancer cell line encyclopedia, or sequence read archives, SRA. These are the regulatories and intellectual property issue that can be arises when you are go through the drug repurposing. Likewise, as per regulatory guidelines, new clinical and or clinical trials may be required to be carried out if the available data are not satisfactory and do not compile with the requirements of regulatory agencies such as FDA, etc. There is no provision of IP protection and of drug discovery by repositioning approach as per IP and patent law. For reposition drug, IP protection is limited. That is the drawback of this repurposing. So selecting the appropriate approach for repositioning is still a challenge due to the regulatory issues, because massive amount of data may not be valid if not obtained from reliable sources. It is, we can say, therefore necessary that researcher or manufacturer must strictly adhere the standard regulatory guideline for drug discovery by repositioning approach. So these are the regulatory and intellectual issues that can be arises if we go through the drug repurposing. At last, the strategic drug repositioning is more systemic and rational way have brought innovation with the discovery of drug molecule with unknown therapeutic indication. I suggest as drug repositioning approach offer insignificant reduction in R&D cost, greater chance of success, shorter research time, and low investment risk has gained increased market demands. The use of in silico technique, that is the AI tool, along with the application of structural based drug design and the pharmacophore modeling strategy and artificial intelligence technology can further accelerate the process of drug repurposing in the drug discovery program. For the betterment of drug repositioning, more in the path of understanding or are required to be executed with integrated approach between the computational experimental method to ensure high success rate of repositioning drugs. So these approaches are defined by me, and I hope have the you can get the information that is going to be useful, and if for further research, and we can also repurpose the any drug for any neglected disease or any kind of disease new indication or any kind of other therapeutic indications. So thank you all. This is all about that repurposing.
That was a wonderful presentation by Deepika ma'am. This was a wonderful and enriching experience for us to hear her views on this topic. We have many more great sessions planned for next few days. So get ready for some fun in next few days. And now last but not least, I would like to thank all the wonderful faculty members and colleagues for attending the FDP program and taking out their important time to attend this session and making it successful. We also looking forward to see you all again in the next session of FDP. Thank you so much, ma'am.
Thank you all. May I end the session?
Yes, ma'am.